This is Dr. Joe Pinter, pediatric neurologist at Oregon Health and Science University. I will be presenting the slides for this chapter for my co-authors, Dr. Harvey Sarnat and Dr. Laura Flores Sarnat, both of the University of Calgary and Alberta Children's Hospital. Table 63.1 lists known gene malformations responsible for several important known CNS malformations. When we are specifically referring in this table to a human gene, we use the convention of denoting the gene symbol in italicized capital letters, but note that because much of our knowledge about human brain development has resulted from searching for human homologs to developmental genes discovered in animals, from Drosophila to mice, we do include in the chapter examples from studies of animals in the discussions of stages, processes, and abnormalities of human neuroembryology. Note that we once thought of brain insults as arising from either environmental or genetic factors, but now recognize these causes are interconnected and inseparable. Environmental factors act by influencing gene regulation and expression, and genetic differences determine responses to environmental agents, including toxins and transcription factors. It's the molecular details of how these molecular genetic pathways are influenced and acted upon by thousands of genes and the proteins they encode that really influence these complex interactions. This figure shows the embryonic vesicles and their adult derivatives schematically in the progression from 3 primary vesicles during the 4th week of gestation just after neural tube formation to 5 secondary vesicles in the 5th week. What is remarkable is that these stages and vesicles were well understood prior to our current understanding of genetics, signals, and processing through the careful work of many different scientists, including O'Rahilly and Muller, and those who developed the original Carnegie stages. This is discussed more in depth in the chapter. Box 63.1 shows many of the important factors in the process of neurulation, which is the bending of the neural placoid to form the neural tube. It requires both extrinsic and intrinsic mechanical forces, in addition to dorsalizing and ventralizing genetic influences, which are discussed in detail in the chapter. Figure 63.2 is a schematic illustration of the formation of the neural tube during the 3rd and 4th weeks of gestation. This figure nicely shows the process of going from a flat 2-layer disc to a 3-dimensional structure, including the neural tube. This lays the structural groundwork for the central nervous system, including the brain and the spinal cord. The neural tube closes in the dorsal midline, first in the cervical region at the anterior neuropore at around 24 days, and the posterior neuropore closes at 28 days. If the anterior neuropore fails to close, this can result in anencephaly, and the posterior neuropore failure to close results in spina bifida with related myelocele or myelomeningocele. Table 63.2 outlines the basic segmentation of the neural tube. Segmentation of the neural tube creates intrinsic compartments that restrict the movement of cells by physical and chemical boundaries between adjacent compartments. These embryonic compartments are known as neuromeres. Hindbrain neuromeres, known by the more specific term rhombomeres, are well established. However, note that although the spinal cord appears to be very well segmented, it is not intrinsically segmented in the embryo or fetus, but rather corresponds in its entirety to the most caudal of the 8 rhombomeres. Interestingly, however, recent genetic expression studies do confirm that there are intrinsic spinal cord segmentation differences that are genetically controlled, and these segments are redesignated myelomeres. Box 63.2 shows programs of important developmental genes, including organizer genes that determine the polarity and gradients of the anatomic axes, and regulator genes that direct the specific differentiation of structures. And in many cases, these regulator genes have served as organizer genes in an earlier period. It is important to note that the difference between an organizer gene and a regulator gene is its function, and the same gene often subserves both roles at different stages of development. Table 63.3 shows several organizer and regulator genes of the embryonic and fetal nervous system, including many with ventralizing or dorsalizing influence. Note also that there is a large family of 38 homeobox genes, also called Hox genes, and each of 13 Hox genes is expressed in certain rhombomeres and not in others. Hox genes are not expressed in the forebrain, and in addition to their functions in establishing compartments of rhombomeres, In the brainstem and affecting differentiation of certain anatomic structures, Hox genes guide the growth cones, forming the long descending and ascending pathways between the brain and spinal cord. Figure 63.3 is a schematic diagram showing the telencephalic flexure in the human fetus at various gestational ages. The telencephalic flexure is the precursor of the Sylvian fissure, one of the 5 fissures of the forebrain. There are, again, 5 fissures, including the interhemispheric fissure, which creates 2 telencephalic hemispheres by sagittal splitting of the prosencephalon at 4.5 weeks gestation. The second is the choroidal fissure at 5 weeks gestation. The third, the hippocampal fissure, which forms after rotation of the hippocampus from a dorsal to a ventral position at 5 to 6 weeks gestation. The fourth is the Sylvian fissure, as noted above, which comes after the telencephalic flexure and is derived at 7 to 8 weeks gestation. And the fifth one is the calcarine fissure on the medial side of the occipital lobe, which forms at 9 weeks gestation. Holoprosencephaly is a relatively common forebrain malformation which results from the non-cleavage of the midline ventral forebrain at approximately 33 days gestation. It is classified neuroanatomically by the degree of failure of hemispheric separation, although there is a poor genotype-phenotype correlation with the 14 recognized genes that can each produce the same or very similar dysplastic morphogenesis. In many cases, it has been associated with trisomy 13, particularly the more severe forms. It is, however, expressed clinically as a spectrum, with the most severe cases termed alobar and the least severe lobar. And there is also another relatively mild variant called the middle inner hemispheric variant. In each of these cases, again, the face often will predict the brain in the sense that more significant facial defects often are related to more significant brain defects, but this is not uniformly true. In this image, the first 2 images are children who have alobar holoprosencephaly. The corpus callosum is absent, and there is no separation at all into hemispheres, and there is a crescent-shaped holoventricle leading into a dorsal cyst. The rightmost image here is a child who has semilobar holoprosencephaly, showing a relatively smaller dorsal cyst and less complete lack of cleavage. Figure 63.5 shows the middle inner hemispheric variant of holoprosencephaly, once thought to be quite rare, but now being seen more commonly with improved imaging technology. Note that the anterior and posterior portions of the corpus callosum are intact and the surrounding anatomy appears grossly normal. Note, however, that there is an absence of interhemispheric fissure and a continuous band of subcortical white matter crossing the midline, and a heterotopic gray matter, see the black arrow above, is seen at the roof of the ventricle as a common finding and gives an appearance of a third caudate head, but it is merely a heterotopic gray matter focus. Figure 63.6 reviews the process of neuroblast proliferation in the ventricular zone. The schematic shows the initiation of migration by asymmetrical cleavages mediated by Notch1 and NUM proteins localized to the basal and apical regions, respectively, of the neuroepithelial cell. Note that within the ventricular zone of the human fetal telencephalon, 33 mitotic cycles provide the total number of neurons required for the mature cerebral cortex. Most mitotic activity in the neuroepithelium occurs at the ventricular surface, and the orientation of the mitotic spindle determines the immediate fate of the daughter cells. If the cleavage plane is perpendicular to the surface, the 2 daughter cells become equal neuroepithelial cells preparing for further mitosis. If the cleavage is parallel to the ventricular surface, then the 2 daughter cells are unequal, and the asymmetrical cleavage results in one at the surface becoming another neuroepithelial cell, whereas the one away from the surface is separated from its ventricular attachment and becomes a postmitotic neuroblast ready to migrate to the cortical plate. In this H&E stain of a coronal section of 16-week fetal forebrain, the germinal matrix and migrating neuroblasts are stained, and one can also see that the sulci are just beginning to develop as marked by the arrows. This schematic diagram shows a cortical neuroblast migrating outward from the ventricular proliferative zone in the cortex along a radial glial fiber. Figure 63.9 shows calretinin immunoreactivity in the normal cerebral temporal neocortex of a 3.5-year-old girl. Calretinin is a marker for GABAergic interneurons that arrive in the cortical plate by tangential rather than radial migration, and they represent about 10% of the total cortical neurons and are distributed in all layers, but predominantly in sensory layers 2 and 4. Figure 63.10 shows gross specimens of a normal fetal brain at 16 weeks gestation that show the presence of only primary sulci, which gives the brain a smooth lissencephalic-like appearance. It should be noted that when looking at fetal MRI, this inability to fully recognize developing sulci is a problem up to 24 and even 26 weeks gestation, so it is difficult to completely accurately prognosticate sulcal maturity based on prenatal imaging at that early stage. Figure 63.11 is a schematic overview of brain gyral development and growth during the embryonic and fetal periods, with the first 5 stages enlarged to show structural detail. It is important, though, to recognize just how incredibly small the brain is at those early stages, because it is easy to recognize when considered that focal insults at those times can lead to large and widespread effects. Lissencephaly, literally meaning smooth brain from the Greek lysos, for smooth, is a severe abnormality of migration. Note in this MRI image, the thin outer cortical layer separated from a deep layer of arrested neurons. The sylvian fissures, noted by the arrows, are open laterally, giving the brain a typical figure of 8 appearance. Figure 63.13 is a coronal T1-weighted MR image showing subcortical laminar heterotopia, which is also known as band heterotopia or double cortex. Note that there are single bands in the parietal lobes, top 2 white arrows, and 2 bands, which are shown by large and small white arrows in the temporal lobes. Note that this is usually seen in girls and is a result of random X inactivation where one of the X chromosomes has a mutation in the double cortin or DCX gene and the other one is normal. Schizencephaly, split or cleft brain, results in a radial defect in migration that results in a peel ependymal seam of abnormal gray matter and either a closed or an open lip defect. In the open lip form seen at the right, gray matter lines the entire cleft and the heterotopic gray matter occurs along the roof of the lateral ventricle. Note that this is easy to mistake, especially on prenatal images, for destructive lesions like porencephaly, and most often postnatal imaging is required to get definitive elaboration of the structural abnormality. Figure 63.15 is a diagram of a coronal section of the cerebral hemispheres of a 3rd trimester fetus or premature infant, indicating 3 sites where lesions such as infarcts or hemorrhages may disrupt radial glial fibers and interfere with neuroblast migration. First, the periventricular region, where radial glial cells may be destroyed. Second, the deep hemispheric white matter, where radial glial fibers may be damaged as they cross. And third, the peel surface, where injury may cause retraction of radial glial fibers and subsequent development of heterotopic collections of neurons. Table 63.4 shows myelination cycles in the human central nervous system based on myelin tissue stains. Table 63.5 shows MRI appearance of myelination based on a table from Barkovich's book. Note that on T1 weighted imaging at birth, the middle cerebellar peduncle and posterior portion of the internal capsule are clearly seen as being myelinated in normal brain, whereas it is very helpful to keep this table so one can review scans of children under 1 year of age to see if the myelination pattern is normal. Note that from birth to 3 months, the pattern on T1 weighted imaging is opposite that of the adult brain, and the adult pattern is not achieved until 2 years of age. It is not clear why myelination changes on T1 and T2 imaging are different. T1 is, again, most useful for assessing myelination in the first year, and in general, handy rules are that myelination progresses from caudad to cephalad, from dorsal to ventral, from central to peripheral, or proximal to distal. The changes in signal reflect mainly increased bound water and myelin, which results in shortening of T1 signal.